Hey guys, welcome back to another Dawn Academy video. As you can see, I'm having a lot of fun with this topic. So we're going to continue 3.5 about cloning because we didn't get to that part because last time was really long. It was too interesting. Okay, so to start off, what is a clone? A clone is, let's start with saying that everyone has identical, not identical DNA, but like identical components of DNA. So like we all have A's, C's, C's, and G's. We all have codons. We all have all that stuff. But we have them in different orders. So we don't all have the same order. We don't all have the same code to make amino acids, the same traits, because we all look different. And even if we looked similar, we wouldn't have identical DNA. We would all have mutations and other things that differentiate us. However, there's this community of people called clones that have identical DNA. They are genetically identical organisms. So examples of these are like the evil twins in the movie that are identical and they hold hands and they're really scary. They have identical DNA because they're identical twins. So basically it's when an egg kind of, when it's a fertilized egg actually, and it keeps multiplying, so it multiplies, we do mitosis, yes. And then we multiply, and these end up forming their own embryo, and these end up forming their own embryo. So they all really start from the same genetic material. So yeah, twins are example A, and we have example B, which is, when we have a plant, yes, of course when we have a plant. When we have a plant, let's say we have this beautiful sunflower, okay? We have this beautiful sunflower, and somehow it gets pollinated, and it makes a plant that looks identical. And then, after that, we have identical trees, and then we have identical fields of identical flowers. That's how all flowers kind of look the same, because they do something called asexual reproduction. Asexual reproduction, which means that they don't do really meiosis. They just keep producing by themselves, or maybe they do meiosis actually. They probably do meiosis, sorry, my bad. But they both are in the same they're in the same flower, so there's not much genetic differentiation. And those are basically clones. They can be considered clones because they're genetically identical. Genetically identical. So yes. Another application example of this is, how starfish, they have this really cool feature where, that's not my finest starfish, but they have this cool feature where if a leg gets cut off, let's say something squishes the leg, oh no, it can make and regrow a genetically identical one. That's just a special talent of starfish. It might look a little wonky, but it's regeneration. So regeneration is also an example of that. It's not making a new organism, but it's similar. Yeah. So there's a lot of different examples of those. So how do we clone cells that aren't from let's say the fertilized egg or from asexual reproduction? What if we have a normal cell that I just take it, we use some pliers, and we just take this little cell out, and it's already differentiated. So let's say it's a heart cell. It might be from your heart. It might be from your liver. It might be from your brain. Like, it can be from anywhere, literally anywhere. But how are we going to make it undifferentiated? Because it already has a function. Either it's a brain cell, or it's a liver cell, or it's a heart cell. How are we going to make it different function? Well, we're going to go through a process called somatic cell nuclear transfer. Let me write that down. So we're going to go through a process called somatic cell nuclear transfer. And the process is exactly like it sounds. We're basically going to take the cell. We're going to make it really simple. Number 1, remove nucleus. Because we don't want the nucleus. We already know that it has a function. It already has the genetic materials. So we want to reset it. We want to completely erase it to 0. We just want the cell. We already have the nucleus though. So we're going to remove the nucleus from this cell, and we're going to throw it into, we're going to take an egg cell. Because, you know, when we're making an embryo, we need a sperm, we need an ova. So we're gonna, when these two fertilize, this one has N chromosomes, right? And this one has N chromosomes too. So when they fertilize and they make this little zygote, this will have 2 N chromosomes. However, how many chromosomes do we have in this differentiated cell? How many? Think of a number in N's. Okay, so because this cell is already developed, it's not an egg or a sperm cell, it is going to have 2 N chromosomes. So once we put it in here, we're going to take an egg cell, remove nucleus. So now we have an empty cell. What do we have? We have one empty cell, one cell, we have one egg, and they're both empty. Empty. And from this, we also have one nucleus, 2N. And here we have one nucleus, which is only N, right? Because it's an egg. So basically, what we're going to do is I'm going to make these into blue. It's going to be blue, it's going to be red. So we're going to take the egg, right? This is our egg. And we're going to put the nucleus from this cell in here with all its genetic material. Let's do them in little chromosomes. So we're going to put all the genetic material in this egg, which is now going to be a 2N egg because of, you know, we have this 2N nucleus from in here. So then at this point, we can make 2 decisions. We can, one, think of kind of like a surrogate. This is a person. My attempt of a person. Oh, that's not too bad. Here's her hand. So this is a surrogate mother. We can choose to either use a surrogate, or, so this is choice surrogate, or we can go for the dish and we can incubate and we can do in vitro fertilization. In vitro fertilization, in vitro is basically when we grow a living organism outside the living organism. So when we're growing life in vitro outside of the organism that it's supposed to grow in. So basically we can take this little egg, we can put it in here, and we have our nucleus, we have our little X's, and we can put it in here and we can incubate it, and we can, you know, heat it up and make sure it grows and it's going to multiply. So after later on, we're going to have more of these. It's going to start multiplying and multiplying and multiplying into clusters of cells. And we're going to end up with quite a few cells. And that all have the same genetic DNA, keep in mind, as this first cell. As this one, sorry, this is our starter cell. We call it starter. Starter cell. The starter cell has the same DNA as all these little minion cells. So they are all really genetically identical. And we can use that word that we learned today, which is clones. They're all identical clones, actually. So that's option number 2. Well, let's go back to option number 1. So option 1 is, we already know that this egg is basically, it acts as a fertilized egg because it has 2 N chromosomes. So we can pretty easily put it back into the surrogate mother, and we can just put it in there, and we can wait for it to grow inside her stomach and kind of make another clone inside there, and ultimately wait for it to grow into an embryo and then have genetic clones. Don't get any ideas, guys. Yeah. So basically that is what somatic cell nuclear transfer is. Yes, said that right. Somatic cell nuclear transfer. That's what it is. We're taking the somatic cell and we're changing the nucleus. We're removing the nucleus and then we're putting it into an egg cell, which will ultimately be a diploid cell, as we can see from over here. It has 2N. And we are going to have two options. We're either going to put it into a dish and grow it in vitro and make a cluster of cells, or we're going to put it into a surrogate mother and watch as the embryo grows into another living organism. So what are some uses of this? We use this for therapy. So cell therapy, whether you have a burn and you need more cells, or if you have maybe leukemia is something for bone marrow transplants, and it can replace other damaged tissues. These are all real life applications, because we can use these cells, and which will ultimately these cells will both, this one especially, will have stem cells, because these cells have no differentiation. So remember when we had stem cells, we had they're basically cells with no function. They're like those people in the group that do nothing, but they're still there. They're cells, but they have no function. And wherever you put them, whether you put them in a heart, whether you put them in an eye, whether you put them in a liver, they'll turn into that organism wherever you put them. So they're extremely helpful, especially for therapy. And yeah, if you guys find out how to do some cloning with simple materials, please let me know. Thanks for watching. If you have any questions, let me know.